Photonic communication within the brain, facilitated by myelinated axons as waveguides, has been proposed as a potential means of neural information transfer. The high refractive index of myelin, approximately 1.45, suggests it could facilitate light guidance and transmission of biophotons, which are photons produced by oxidative processes in living cells. Biophotons have been observed in various organisms and tissues, including the brain, and their spectral range is influenced by species and aging processes. Research has shown that biophotons are produced in the brain and their exchange between neurons is proposed as a means of information transfer, with the possibility of encoding information through biophoton production. Preservation of polarization in light transmission along axonal connections is a crucial aspect for any form of photonic communication. Using simulations of guided mode propagation through a myelinated axon structure with multiple Ranvier nodes, the authors find that polarization can be well preserved through multiple nodes. The simulations model the transmission of light through a series of nodes, each consisting of a myelinated segment and a node of Ranvier, where the myelin sheath is interrupted. The results show that transmission losses through multiple nodes are approximately multiplicative, indicating that the total loss is proportional to the product of the losses at each node. These findings strengthen the hypothesis of neural information transmission facilitated by biophotons, supporting the possibility of both classical and quantum photonic communication within the brain. The study contributes to the understanding of higher-order functions such as learning, memory, and consciousness, which are still not fully understood in neuroscience. The correlation between biophotons and neural activity, as well as their association with reactive oxygen species, highlights the potential role of biophotons in cellular communication. Overall, this research provides an important context for the hypothesis of photonic information transfer in the brain, advancing our comprehension of neural processing and communication mechanisms. In this section of the research paper, the authors delve into the potential for myelinated axons to serve as biophoton waveguides, a concept that has garnered significant attention in recent years. They discuss how the dynamic nature of myelination and its effects on axonal properties could impact light transmission. The paper highlights previous studies demonstrating the feasibility of myelinated axons as waveguides, despite optical imperfections, and experimental evidence supporting light conduction along white matter tracts. The authors then explore the unique features of photonic communication in the brain, including the potential for quantum communication due to photon polarization. It emphasizes the importance of assessing the effective transmission of biophotons and their polarization in the brain environment as a biologically realistic phenomenon. The paper outlines the author's approach to studying guided mode propagation through a myelinated axon model, focusing on polarization evolution and transmission across multiple Ranvier nodes. This is the first study to consider the evolution of light polarization in the myelin sheath. The paper details the methods used, including the axon model and software, and presents findings on polarization evolution and transmission of a guided mode through the model with 4 Ranvier nodes. The implications of these results, suggested improvements to the model, and potential applications are discussed. Additional calculations for other modes of the myelinated axon are included in the appendix, along with supplementary figures illustrating polarization evolution in the myelin sheath. A myelinated axon model is constructed using ANSYS numerical FDTD, 3D electromagnetic simulator software, comprising 3 fundamental materials: axon, myelin sheath, and interstitial fluid. These materials are modeled as uniform dielectric materials with specified refractive indices, consistent with biological observations. 1.38 for the axon, 1.44 for the myelin sheath, and 1.34 for the interstitial fluid. The myelinated axon is roughly cylindrical in shape, with an ideal cylindrical structure featuring an axon radius of 0.6 mm and a myelin radius of 1 mm. Resulting in a myelin thickness of 0.4 mm. This design ensures good confinement of light within the myelin sheath while maintaining manageable computational demands. The model also incorporates Ranvier nodes, segments of bare axons without a myelin sheath, which are critical for assessing the wave-guiding ability of the myelin sheath. These nodes are approximately 12 mm in length and present a significant opportunity for transmission loss. In the model, The Ranvier node length is set to 2 mm, and the node-etanode distance is set to 100 mm, resulting in an internode length of 98 mm. The model spans 500 mm in length and includes 4 Ranvier nodes. Paranodal regions, located at the boundaries between internode and Ranvier node segments, are not modeled due to complexity limitations. The assumption of low biophoton absorption in the myelin-sheath is supported by studies on brain tissue, where absorption in white matter is relatively low and primarily due to scattering rather than absorption. This model enables the simulation of light propagation in a myelinated axon, allowing for the investigation of the optical properties of this complex biological system. The internodal region of a myelinated axon model is illustrated in Figure 1, showcasing the axon, myelin-sheath, and a test simulation region. The cross-section of the internodal region is annotated with the axon radius ρ, outer myelin radius erm, and myelin thickness T. To construct models and run simulations, the project utilizes ANSYS numerical FDTD, 3D electromagnetic simulator software, which employs the finite difference time domain method to solve Maxwell's equations for models on the nanometer scale. This enables the calculation of modes and simulation of light propagation in the myelinated axon model. The FDTD method solves the electric and magnetic field components on a discrete spatial and temporal grid within the Yee cell paradigm. The simulation region is set to span 4 mm in the x and y directions, with a length of 500 mm in the z direction. A planar source is applied as an input mode at the beginning of the region at z equals 0 mm. 20 cross-sectional monitors are placed along the z-axis to collect transmission and electric field profile data, with a spacing of 25 mm apart, starting at z equals 25 mm and ending at z equals 500 mm. The simulation results show the electric field profile, both magnitude and polarization, at different cross-sections throughout the structure. The electric field behavior is observed well after any given Ranvier node, and transmission as a function of distance along the myelin sheath is presented in a log plot. The overlap of the complex electric field cross-sections with the input mode is also presented as a function of distance along the myelin sheath. Consistent with previous studies, mode calculations for the current model support the existence of modes in the myelinated axon structure. The first 6 modes calculated for the myelinated axon model are presented, validating the accuracy of the simulation approach. In a myelinated axon structure, the transmission of guided light is significantly influenced by Ranvier nodes, as demonstrated by this research. A 4-node model was used to simulate light transmission, with an input mode, HEO21, characterized by 4 high-magnitude antinodes interspersed with low-magnitude nodes and a hyperbolic polarization pattern. The simulation results show that the input mode remains confined to the myelin sheath throughout the structure, with the hyperbolic polarization pattern well preserved. The transmission coefficient, T, was recorded every 25 mm along the z-axis and plotted against Z, revealing perfect transmission prior to the first Ranvier node, as expected. However, noticeable decreases in transmission occur at each subsequent Ranvier node, indicating some loss due to these nodes. The results show slightly higher transmission than expected, compared to the assumption of exponential transmission drop-off with each Ranvier node. To further analyze polarization transmission, the normalized overlap of complex electric field profiles at various points with the input field profiles was calculated, providing a measure of polarization transmission fidelity, F(Z). This metric reveals how well the polarization state of the input mode is preserved as it propagates through the structure. The findings suggest that while the hyperbolic polarization pattern remains well preserved, the transmission of light is affected by the presence of Ranvier nodes. This study highlights the importance of considering the role of Ranvier nodes in the transmission of guided light within a myelinated axon structure. Further research is needed to fully understand the implications of these results for neural signal transmission and potential applications in bio-inspired photonic devices. The fidelity function f(z), defined in equation 1, is a crucial measure for understanding the behavior of the propagating mode as it traverses the Ranvier nodes. It represents the inner product of 2 complex vectors and is calculated as the ratio of the inner product of the electric field at position z and the electric field at position 0, to the inner product of the electric field at position 0 and itself. Mathematically, this can be represented as F, Z, equals integral RH tilde E, x, y, 0, tilde E, x, y, Z, I dx dy, integral RH tilde E, x, y, 0, tilde E, x, y, 0, I dx dy, integral RH tilde E, x, y, Z, tilde E, x, y, Z, I dx dy, where h, I denotes the inner product of two complex vectors. The results of this calculation are visualized in Figure 5, which shows oscillations in the fidelity function, likely caused by the mixing of lossy modes at the Ranvier nodes. These oscillations interfere with the propagating mode, but as expected from the mode profiles in Figure 3, the fidelity generally remains high at all points. Notably, the fidelity returns to values very close to 1 after the fourth node, indicating that the propagating mode is well preserved despite the presence of lossy modes. This observation is consistent with the visual inspection of the mode profiles in Figure 3, which suggests that the propagating mode is robust against the mixing of lossy modes. The fidelity function provides a quantitative measure of the mode's robustness, allowing for a more detailed understanding of the underlying physics. Figure 3 presents the electric field profiles of the calculated input mode He11 and cross-sections of the 4-Ranvier-node myelinated axon structure at positions 75, 175, 275, 375, and 475 mm. The mode source is located at 0 mm, with an input amplitude of 100 volts per meter. The Ranvier nodes are 2 mm long and begin at Z equals 100, 200, 300, and 400 mm. The plots in Figure 3 are organized into two columns, with the left column, A, C, E, G, I, K, depicting the electric field magnitude, and the right column, B, D, F, H, J, L, depicting the electric field vectors. The black arrows in the right column plots represent the normalized three-dimensional electric field vector at a given position, while the red arrows represent the magnitude-scaled three-dimensional electric field vector at a given position. The color-coded contour lines in the plots depict the magnitude of Ez, with electric field magnitudes given in units of V per meter. The electric field profiles and cross-sections of the myelinated axon structure are presented to illustrate the spatial distribution of the electric field at different positions along the axon. This figure provides a visual representation of the electric field behavior in the vicinity of the Ranvier nodes, which is crucial for understanding the underlying mechanisms of electrical signal propagation in myelinated axons. This figure builds upon the previous pages, which introduced the theoretical framework and simulation setup, and provides a foundation for the subsequent analysis and results presented in the paper. The transmission coefficient of a myelinated axon structure decreases exponentially with distance along the axon, as depicted in Figure 4. Measured from the mode source at Z equals 0 mm, Ranvier nodes, each 2 mm long, are located at Z equals 100, 200, 300, 400 mm. The blue points in the figure represent transmission data collected from the simulation, while the green points represent expected transmission data under the assumption of exponentiated loss through each Ranvier node, using the measured transmission loss from the first Ranvier node. The polarization transmission fidelity, F(Z), is presented in Figure 5 as a function of longitudinal position Z. Defined in EQ. 1, F(Z) indicates how well the polarization state of the mode is preserved as it propagates along the axon. The graph shows that F(Z) decreases with increasing distance, suggesting a degradation in the preservation of the polarization state over longer distances. These findings provide insight into the behavior of light transmission within myelinated axon structures, highlighting the impact of Ranvier nodes on transmission properties. Crucially, these results are essential for understanding the optical properties of such structures and their implications for potential biomedical applications. Research on guided light propagation in the myelin-sheath with multiple Ranvier nodes has been conducted using ANSYS numerical FDTD, 3D electromagnetic simulator. The model incorporates biologically realistic structural and optical properties for axon, myelin-sheath, and interstitial fluid material. Simulation results demonstrate that the input mode's polarization state remains well preserved throughout the 4-Ranvier-node myelin-sheath model, exhibiting high polarization fidelity between the electric field and the input mode within the myelinated axon model. This finding suggests potential applications in biophotonic quantum communication, offering a possible solution for addressing the brain's high computational demands under energy constraints, particularly for functions such as consciousness. Key results include the preservation of the input mode's polarization state, which is critical for maintaining signal integrity in quantum communication systems. The study's limitations include the assumption of a non-point-like nature of biophoton sources and the absence of a detailed description of how these sources couple into the modes of the myelin sheath. Future research directions include investigating the polarization evolution across realistic ranges of biological parameters and assessing the impact of optical imperfections on light propagation within the myelin sheath. Understanding light interactions with the myelin sheath can also contribute to advancements in neuroimaging, nanoscale optogenetics, and a deeper understanding of diseases associated with the myelin sheath. Further research in this area can provide valuable insights into the complex interplay between light, matter, and biological systems, ultimately leading to innovative solutions for addressing the brain's computational challenges. This research paper presents a detailed compilation of findings, methodologies, and implications in the field of computational biology. The primary focus is on the development of novel algorithms that enhance our understanding of genetic sequences and their role in disease susceptibility. The paper begins by outlining the importance of computational tools in deciphering complex genetic information. It highlights how these tools can identify patterns, predict potential mutations, and provide insights into evolutionary processes. The authors emphasize the need for more sophisticated algorithms to tackle the vast amounts of data generated from high-throughput sequencing technologies. A significant portion of the paper is dedicated to discussing various computational models used in genomics research. These include hidden Markov models, which are statistical models used to analyze sequences of observations, and machine learning techniques such as random forests and support vector machines, which are applied to predict gene function and identify potential drug targets. The authors also delve into the concept of pangenomics, an approach that considers the complete gene repertoire of a species rather than just a single reference genome. This allows for a more comprehensive understanding of genetic diversity and its relationship with disease susceptibility. Furthermore, the paper touches upon the ethical considerations associated with genomic research, particularly concerning privacy and consent. It emphasizes the need for transparent data handling practices and strict guidelines to protect individual privacy while advancing scientific knowledge. In conclusion, this research paper offers a comprehensive overview of the current state of computational biology, highlighting both the achievements and challenges in this rapidly evolving field. It underscores the importance of continued innovation in algorithm development, data analysis, and ethical considerations to ensure that genomic research benefits society without compromising individual rights. Reactive oxygen species play a crucial role in synaptic plasticity and memory, as evidenced by studies highlighting their importance in these processes. Specifically, cysteine oxidation by reactive oxygen species regulates intracellular signaling. Furthermore, photons guided by axons may enable backpropagation-based learning in the brain, a concept with significant implications for our understanding of neural communication. Myelination is another critical factor in brain function, with conduction velocity directly related to the degree of myelination. Muller cells, acting as living optical fibers in the vertebrate retina, also contribute to the regulation of light-based communication. Synchronized myelin changes, in turn, shape brain function and plasticity. Simulations of nerve fibers based on anti-resonant reflecting optical waveguides have shed light on the guidance of electromagnetic waves by myelinated axons in the optical and infrared ranges. The node of Ranvier has been conceptualized as an array of bionanoantennas for infrared communication in nerve tissue. Electromagnetic characteristics of in vivo nerve fibers at the terahertz far infrared band have been studied, as well as photonic transmission inside brain nerve fibers. Antiresonance, inhibited coupling, and mode transition in depressed core fibers have been examined, along with anisotropic light scattering from myelinated axons in the spinal cord. The dependence of light propagation in the brain on nerve fiber orientation has also been explored. Intriguingly, the authors suggest that quantum physics may hold the key to solving the hard problem of consciousness. Finally, studies on topography and refractometry of nanostructures using spatial light interference microscopy, as well as light propagation in tissues with controlled optical properties, have been reviewed. These findings collectively underscore the complex interplay between light, cellular communication, and brain function, offering new avenues for research into the mysteries of neural communication and consciousness. Optical properties of mammalian brain tissue, a comprehensive review. The interaction between light and neural tissue is a multifaceted phenomenon that has garnered significant attention in recent years. This review delves into the absorption coefficients of mammalian fat, the effects of light on neural function and metabolism, and the potential role of quantum effects in the brain. The absorption coefficients of mammalian fat are crucial for understanding how light interacts with neural tissue. These coefficients vary across different species and can be influenced by factors such as wavelength and temperature. For instance, studies have shown that the absorption coefficient of rat fat decreases with increasing wavelength, indicating a higher absorption of shorter wavelengths. Light's interaction with neural tissue is complex and multifaceted. Cytochrome oxidase, a protein found in mitochondria, has been identified as a possible photoreceptor that could play a role in regulating metabolic rate and synaptic efficacy in cortical neurons. This interaction may be critical for understanding the neural response to light exposure. Light also has a profound impact on neural function, particularly in the context of functional connectivity at rest. Studies have demonstrated that light can alter functional connectivity in the brain, suggesting a potential role in modulating neural activity. Additionally, light can suppress thermogenesis by activating opsin-5 hypothalamic neurons, which could have implications for energy homeostasis and overall health. Quantum effects in the brain are an emerging area of research, with theories suggesting that quantum cognition may be possible through the manipulation of nuclear spins. This concept is still speculative, but it presents intriguing possibilities for future investigations into the neural basis of consciousness and cognition. In addition to these topics, the optical properties of vertebrate nerve axons are critical for a comprehensive analysis of neural function. Axonal size and myelination can significantly influence the speed at which neural signals propagate, making them essential components of neural communication. Understanding these properties is therefore paramount for elucidating the intricacies of neural function and behavior. This review highlights the complex interplay between light and neural tissue, emphasizing the importance of continued research in this area. By elucidating the optical properties of mammalian brain tissue, scientists can gain a deeper understanding of the neural mechanisms underlying perception, cognition, and behavior, ultimately paving the way for novel therapeutic interventions and technological advancements. Transmission of electromagnetic waves in soft material waveguides is a complex phenomenon that has garnered significant attention in recent years. This research endeavors to elucidate the fundamental properties of these transmissions, leveraging both experimental and computational methodologies. In the realm of experimental approaches, 3D polarized light imaging is employed to visualize fiber architecture in the brain, providing high-resolution insights into the intricate structures that govern wave propagation. Complementing these experimental findings, computational models are developed to simulate the behavior of electromagnetic waves in soft material waveguides. A crucial aspect of this research is the characterization of waveguide dispersion, which is achieved through the implementation of a novel algorithm that accurately captures the modal properties of the waveguide. This algorithm is rooted in the solution of the vectorial Helmholtz equation, which is discretized using a finite element method. The study reveals that the dispersion characteristics of soft material waveguides are profoundly influenced by the material properties and geometrical configurations of the waveguide. Notably, the results demonstrate that the waveguide dispersion can be tailored by judiciously selecting the material properties and waveguide geometries. The findings of this research have significant implications for the development of novel biomedical devices and sensors that exploit the unique properties of soft material waveguides. Furthermore, this study lays the foundation for future investigations into the application of soft material waveguides in various fields, including optics, electromagnetics, and biomedicine. The authors investigate the modal properties of circular isotropic optical fibers by calculating the effective index and electric field distribution for each mode. The electric field is well confined to the myelin-sheath, exhibiting alternating bright and dim areas, referred to as antinodes and nodes, respectively. Due to degeneracy, these modes can be sorted into pairs related by a simple rotation. 6 calculated modes are presented, following established convention. HEO11 and HE11 modes have linear polarization patterns and an effective index of 1.4412, with 2 antinodes and 2 nodes related by a 90-degree rotation. The HEO21 and HE21 modes exhibit hyperbolic polarization patterns, with an effective index of 1.4082, and 4 antinodes and 4 nodes related by a 45-degree rotation. In contrast, TM01 and TE01 modes exhibit cylindrical symmetry and are non-degenerate, with TM01 having azimuthal polarization and an effective index of 1.4090, and TE01 having radial polarization and an effective index of 1.4071. These modes show magnitudes equally distributed through the ring of the myelin sheath without antinodes or nodes. The electric field magnitude plots and vector plots, presented in Figure 6, display the spatial distribution of the electric field for each mode. Notably, the EZ component of the electric field is negligible in all cases, on the order of 10 to the power of 17 V per meter or less. The input light amplitude is 1 volt per meter, and the plots are calculated over the cross-section of the model at the source. Z equals 0 mm, providing insight into the modal properties of the circular isotropic optical fibers.